A very good morning to all. A closer look of the anti-VEGF injections. It began as early as in 1948 when Michelson had hypothesized that ischemic retina released a factor X. It was in 1983 when Senger et al. From Boston identified the molecule and named it as the vascular permeability factor. Ferreira et al. In 1989 identified this molecule as vascular endothelial growth factor, a major regulator of angiogenesis. Further publication in 1989 of two back-to-back articles in Science began a new phase in this chronicle, the one that culminated in the development of the latest anti-angiogenic therapies. 1994, the American Journal of Ophthalmology published a key article showing that the hypoxic retina produces VEGF, suggesting the role of VEGF in ocular neovascularization. A study was conducted by lasering the non-human primate eyes, inducing neovascularization, which induced neovascularization of the iris. So this showed that a report of elevated levels of VEGF in the ocular fluids of patients with active neovascular ocular disease. So in this presentation, we go through the VEGF molecule, the VEGF receptor structure, how the VEGF signaling happens, and how is the anti-VEGF therapy useful. The vascular endothelial growth factor, also known as the vascular permeability factor, was originally described as endothelial cell-specific mitogen. Besides endothelial cells, it is being produced by other cell types, including tumor cells, macrophages, platelets, keratinocytes, and renal mesangial cells. So what is a vascular endothelial growth factor? It is basically a homodimeric glycoprotein which belongs to a family of secreted polypeptides. It is highly conserved receptor binding cystine knot structure. This shows the structure of a VEGF molecule. Which are the sources of VEGF molecule in the eye? In the retina, it is being produced by the Muller cells, the endothelial cells of the vessels or the supporting pericytes, the retinal pigment epithelial cells, and the retinal neurons and the astrocytes. And in the anterior segment, we have the non-pigmented ciliary epithelium, which is considered to be the reason for the ocular neovascularization of the iris. VEGF-A molecule is considered to be the founding member of the family, the single most important regulator of blood vessel formation, and is essential for embryonic vasculogenesis as well as angiogenesis. It is the key mediator of neovascularization in cancer as well as in other diseases. So how do the VEGF molecules exert their action? The hypoxic cells produce the upregulation of the VEGF molecules. There are two main mechanisms which happens. One is by the induction of angiogenesis, and secondly by the induction of an inflammatory process. The induction of hypoxia-inducible factor 1, and the consequent transcriptional activation of target genes. Increased VEGF transcription causes upregulation of angiogenesis and thereby restoring the oxygen supply and the nutrition supply of the hypoxic tissues. And how do the inflammatory process works? It acts by inducing the expression of vascular cell adhesion molecules, molecule 1, and enhancing the leukocyte recruitment and endothelial cell adhesion. This increases the blood-retinal barrier breakdown. So there are two basic mechanisms in how the VEGF molecule acts. One is by induction of angiogenesis, and secondly by induction of blood-retinal barrier breakdown. So just like VEGF-A being the prototypical member of the family of a related growth factors, it also includes placental growth factor, VEGF-B molecule, VEGF-C molecule, the VEGF-D molecule, also known as the force-induced growth factor, and VEGF-E, which is considered to be the viral factor. And another interesting variant of VEGF is also expressed in the venom of habu snake. It's a variety of pit viper native to Japan. VEGF molecules are now mediated by a family of protein tyrosine kinase receptors. So it has basically three structural importance. One is three parts. One is an extracellular portion consisting of 7 immunoglobulin-like domains. Secondly, you have a single transmembrane spanning region. And thirdly, there is an intracellular portion consisting of a split tyrosine kinase domains, which is of most important part of the VEGF receptor. So where are the VEGF receptors located? They are in the endothelial cells, retinal pigment epithelial cells, as well as in the bone marrow derived epithelial cells, which is of importance in cancer patients. So what is the difference between vasculogenesis and angiogenesis? Vasculogenesis, it is the formation of new blood vessels from angioblast. Angioblasts are the endothelial precursor cells which differentiate into endothelial cells. And what is angiogenesis? That is a growth of new blood vessels from already existing blood vessels. That is more of a pathological change. So this flowchart shows how which all VEGF molecules act in the corresponding VEGF receptors. Here we can see that the VEGF, the name VEGF corresponds to VEGF A molecule. It acts via the VEGF receptor 1 and the VEGF receptor 2, and the action mediated via the VEGF receptor 1 is mainly vascular organization. That is the expression of matrix proteases and thereby stabilizing the vessels. VEGF receptor 2 mediates mainly angiogenesis, that is the proliferation of the new vessels. The VEGF C and D molecules act via the VEGF receptor 2 and VEGF receptor 3, and the action mediated mostly through the VEGF receptor 3 is lymphangiogenesis. So these are the main receptors which promote the action of the VEGF molecules. And certain subtypes of VEGF A are VEGF 165, which is most important, and VEGF 145. And they tend to act via the co-receptors, which have the efficacy only half of that of the main VEGF receptors. They are called neuropilin receptors. So they promote angiogenesis, as we have seen, and vascular permeability, cell migration, and gene expression. And Lee et al. Had proved that there is an autocrine loop for VEGF A molecule, which contributes mainly to the endothelial functions. So when VEGF molecule binds to the VEGF receptors, it initiates a cascade of events. There are two main mechanisms. One is by activation of a phospholipase and phosphokinase pathway, which is being which is being the main mechanism for most of the anti-VEGF drugs. And there is another AKT pathway. AKT pathway is the main mode of action of the tyrosine kinase inhibitor drugs. So it causes a balance of the pro and the anti-angiogenic signals, resulting in sprouting of the new blood vessels, cell proliferation, and cell migration. So when there is a VEGF receptor signaling, VEGF receptor 1 has a higher affinity for VEGF A molecules, and the tyrosine kinase activity is approximately 10-fold weaker than that of a VEGF receptor 2. The major pro-angiogenic signal is believed to be generated from the ligand-activated VEGF receptor 2. There is a negative regulatory role for VEGF receptor 1 in angiogenesis at early embryogenesis. So the anti-VEGF antibodies, the efficacy is basically in terms of their vascular normalization. Once an anti-VEGF antibody is proved to be effective, it makes the abnormal vessels well covered with pericytes and thereby lowering the vascular permeability. The anti-VEGF receptor drugs have been classified into two main groups. One is of importance to the ophthalmologist, as we all know, the anti-VEGF antibodies. And secondly, the tyrosine kinase inhibitors like imatinib, sunitinib, which are of importance to the oncologist. And certain other groups include the VEGF trap molecules, soluble VEGF receptor peptide vaccines, and antiplatelet growth factor antibodies. And these are, most of them have come into market, like VEGF trap is the aflibercept, and rest are undergoing preclinical and the clinical trials. So it is that the area of ischemic retina, which characterize a number of ocular pathologies, most notably in ROP and diabetic retinopathy, which increase the need for the development of the anti-angiogenic therapies. And the first treatment developed using this VEGF neutralizing strategy was bevacizumab for systemic use, mainly for the purpose of cancer treatment. And secondly, of importance to the ophthalmologist is the neovascular AMD. The first drug approved by the FDA was pegaptinib, an RNA aptamer that binds and neutralizes VEGF 165. And the drugs will be dealt in detail in the upcoming presentations. So after 6 full decades, Michelson proposed the existence of factor X molecule. We are right now standing in an era of anti-VEGF molecules which have been adopted and proven to be valuable in the treatment of a lot of blinding eye conditions. Thank you.
Thank you, Anu, for that detailed presentation.